Okay, we're back for the last little podcast for Area Study 1. This one's on cellular respiration, the other major chemical equation we need to be aware of. So again, it has a purpose, it has a very, very important purpose. Without cellular respiration, all that photosynthesis is going to waste. This is that bit of physics you really need to be aware of. Light energy arrives on the planet from the sun, more than we can possibly use every day. Plants convert about 10% of it into stored chemical energy. But then, somehow, we have to turn it into a form of energy, and we use the currency as ATP for living things, into forms of ATP. So we've got to go from light energy to chemical stored energy to energy of action. So, cellular respiration takes place in all organisms. We either have, well, in the case of plants, have made the glucose, now they need to break it down to release the energy, or in our case, we eat the plants or eat animals that have eaten plants to get hold of that carbohydrate, break it down into glucose, and then break the glucose down again. So it takes a bit longer for us. And all organisms, every single living thing, uses ATP, the energy carried in ATP, in the phosphate bonds, as a currency, as that source of energy. So critically important for all living things. And we've got a chemical equation that you need to memorize. 1 glucose is oxidized by 6 oxygen molecules to produce 6 carbon dioxide molecules, waste product, 6 water molecules, another waste product that you will know as metabolic water. This is a wastewater from our metabolism that some animals actually get by on, and energy in the form of adenosine triphosphate. And there's a lot of discussion about how much actually gets produced, and the reality is that 38 would be probably maximum efficiency, and few animals or plants ever work at maximum efficiency, but potentially between 36 and 38 units of ATP. And cellular respiration occurs in 3 stages. Glycolysis, outside the mitochondrion, in the cytosol, Kreb cycle, and then electron transport chain. And these both occur within the mitochondrion and produce in the end most of the ATP. And you need to know locations, inputs, outputs, and the amount of energy produced at each. You're going to be examined on it. So the mitochondrion is also believed to be an ancient prokaryotic organism living in symbiosis with eukaryotes. And again, they have a double biphospholipid layer. They have DNA occurring in plasmid loops like prokaryotes do. And again, they have the ability to self-replicate. So in my heart and other muscles in my body, I have lots of mitochondria. In my adipose tissue, I have very little. That's why adipose is quite hard to break down. The mitochondria aren't there to use them. So the development of adipose, I know all about adipose, it's very hard to get rid of. So the first stage is glycolysis. This occurs in the cytosol of the cell. And glycolysis literally means splitting, lysis, of a glucose molecule. Glucose, lysis, split. You see lysis a lot. 1 glucose molecule is split to create 2 pyruvate molecules. This also produces 2 NADH hydrogen acceptors, so we've got a way of carrying some hydrogen, and 4 molecules of ATP. 2 of these will be spent crossing into the mitochondria. So as a process, it's not producing a lot of ATP. There's ATP already. So if there's adequate oxygen, so if we're breathing nicely and we're alive and not buried in dirt somewhere, then the process will continue on into the mitochondrion. Otherwise, we'll go into anaerobic respiration, which we'll talk about after we've talked about Krebs and electron transport. So Kreb cycle, those 2 pyruvate and the 2 NADHs move into the mitochondria in the cost of that ATP, 2 ATP. The pyruvate and NADH are transformed using 2 acetyl coenzyme A molecules, and by doing that they create 6 NADH, 2 FADH2, and 6 CO2 molecules. So the wastes have already been produced, we get rid of the CO2, and we get 2 more units of ATP. Not a great deal of ATP, but we've set the chemicals up. These NADHs and FADHs carry the electrons into the electron transfer stage. So In electron transport, the NADHs and the FADHs release their electrons. These combine with these hydrogens through the ATP pump and load ADP with a phosphorus group to create ATP. And those ATPs are containing that energy formed for the cell. And this can when it's working perfectly well, produce up to 34 molecules of ATP. And the rest of our waste, the water. And that's as simple as that, isn't it? Well, of course, as you can imagine, it's incredibly more complex than that. And we'll look at the complexity in class using a couple of podcasts from my good friend Paul Anderson at Bozeman Biology. But essentially, that's what you need to know. The other process is known as anaerobic respiration. And there are, of course, many organisms which use anaerobic respiration and survive in very low oxygen environments because of that. Plants and bacterium are particularly good at anaerobic respiration without killing themselves, and they result in toxic wastes, alcohol and CO2. And of course we use that in the production of alcohol for our consumption. We use that in the creation of breads. The CO2 is what we use to create the fluffy bread. We create cheeses using this process. There's a whole lot of things we can do with this to create foods and drinks for ourselves. So it's very useful. In animals, we get lactic acid and CO2 buildup. And these are both deleterious to our organism. They actually create problems. And many will have cramped after exercise. That's often because of a lactic acid buildup. I'm always astounded to watch the guys and women who've just had a big race in the pool, and they hop in the cool down pool. Or they've done the sprint around the stadium, and they go and have another run. Light run, just to burn off that lactic acid which is built up. It's light exercise, it's just using the lactic in the presence of oxygen, because they've created a whole lot of it. Again, there are limiting factors on the ability of cell respiration to occur. Temperature is one of those. It requires enzymes to operate, and of course, high temperature will stop our enzymes operating because they get denatured. At low temperatures, they work more slowly. Oxygen is obviously important. We need oxygen to combust, to oxidize the glucose. And so in low oxygen environments, we're going to see anaerobic respiration for short periods while we deal with, get life happening and then deal with the waste later. And obviously you need glucose. No glucose, no cell respiration. Low glucose, low energy levels. High glucose, well, we start storing it away, don't we? We store it essentially we store it as glycogen, plants store it as starch, and in humans, of course, and other animals, that becomes adipose stools. And there we go. That is Area Study 1 in a nutshell. We'll talk much more detail in class clearly.